In this video, video 20, we'll describe with the aid of diagrams and photographs the distribution of xylem and phloem tissue in roots, stems and leaves of dicotyledonous plants. We'll also describe with the aid of diagrams and photographs the structure and function of xylem vessels, sieve tube elements and companion cells. Now, in humans, when you look at veins and arteries, we tend to find single veins and single arteries running to different parts of the body. In plants, this does not happen. The vessels tend to be very small, and there tends to be very many of them in what we call a bundle. So rather than having simply one vessel like that, you tend to have a bundle of different vessels joined together, and these are called vascular bundles. And that's obviously a side view. If I then cut that diagram in half, like so, and then turned it 90 degrees facing you to show a cross section, you would see a bundle looking a little bit like this, and that's called a vascular bundle. So write that down, vascular bundle. Now, part of getting a good grade in biology, we're talking a C or above, is learning the sheer number of vocabulary or words in the subject and what they mean in detail. Of course, you have to apply these words, understand them, but before you can do that, you really must learn them. You can use a variety of ways to learn them: flashcards, diagrams, mind maps, and so on. But the key is to make sure you know off by heart all of the words that are necessary to answer the question, because quite simply, if you've got long answer questions, you cannot score marks without using the appropriate terminology correctly, unlike GCSE. So first of all, let's have a diagram, or a photograph even, of the vascular tissue within a root. So within the root, you get the following pattern. You first of all have the xylem stained in green, there, here, there and there, and it kind of forms a cross or an X. And the reason why it forms this shape is it's structurally solid. So as the roots grow, they have pulling forces on them, and this deforms the root. And of course, if it wasn't for the fact that these xylem vessels had a strong structure, they could risk breaking apart, and the individual xylem are more vulnerable. So the cross shape provides that structure to the pulling forces. And then you see these dark red patches there, 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 and there between the arms of the xylem. You might call them arms, or the ends of the crosses. And those dark circles are phloem. So you should remember the xylem transports water and dissolved minerals, and the phloem transports sugar, such as sucrose, through the plant. And in the roots, of course, you won't find much sugar being produced. And you're obviously aware that the roots are underground, so photosynthesis will not really be happening too much when there's no light. And also, they don't have huge access to carbon dioxide unless it's transported in the plant. So there's no production of sugar in the roots, but the roots still need sugar. Can you think of why this would be? Well, actually, the roots are made of cells. Cells all must respire to release energy for the jobs they do, the metabolism they go through, and therefore the roots need to be supplied with sugar, and they're called what we call a sink. So a sink, S-I-N-K, is a place where the sugar flows to, and a source would be where the sugar is produced from. And of course, the leaves produce sugar, and the roots would need the sugar. So usually we say that the roots act mostly like a sink. And we also know that the roots carry water up the plant, and that's why it has a bundle of xylem here to allow water through, and we'll discuss that later on in more detail. So we've done the roots, and we understand the structure of the xylem and the phloem in the roots, and we'll now look at the xylem and phloem in the stem. So this diagram shows you the structure of the xylem and phloem in the stem. And this is actually quite zoomed in, it's times 100, so you can imagine that it's only a very small portion of a sunflower stem, and actually this tends to go around like that in a huge circuit, which I can't show on this current diagram. And the vascular bundles of xylem and phloem are actually found on the outer edge of the stem, so near the surface of the stem, not deep inside. In non-woody plants, so plants without a lot of wood like bark, the bundles tend to be discrete layers and separate, so you'll find a bundle there, a bundle there, a bundle there, with quite big gaps in between. Whereas when you find woody plants, the bundles tend to link together continuously, so you see these really strong ridged patterns. And that complete ring of vascular tissue running around the larger woody plants means that there's a very strong and flexible property of the wood and the branches and the stems, and they can withstand the forces they undergo when things like wind blows the branches. Now, the xylem is actually found on the inside, so the xylem is found here. The phloem is found on the outside of each vascular bundle. The phloem is found in the pinky bit there. And between the xylem and the phloem, there's actually a band there, and that band's called the cambium. So I'm going to draw this on the board in a second. So, I'll draw an arrow there, an arrow there, and an arrow there. So, we should know that this is the xylem. There we are. This is the phloem. And of course, we've got the cambium. Now, the cambium divides the xylem and phloem. And the cambium is actually made of cells called meristem cells. Now, meri in Latin means divide or division. So the meristem cells divide the stem, and those are found in the cambium, of course. I'll put some brackets there so you don't get confused. And basically, the meristem cells are essentially like stem cells, as you would guess, and they divide, and they can differentiate into xylem or phloem. So we call the meristem cells pluripotent because actually they can divide into a number of different things. Well, namely, they can divide into xylem or phloem vessels. Pluripotent is different to totipotent or omnipotent because it doesn't divide into any cell in the plant at all. It just divides into mainly those two specific cells, which are xylem or phloem. Now, there will be a lot of C words in the module. There are some words that sound a bit like cambium, but you need to remember the cambium is actually found between the xylem and phloem in the vascular bundle. Now, I'm going to move this out of the way. I'm going to quickly go back to the root. We've covered the stem in detail, and I should probably touch on the fact that around the outside of the vascular bundle, there's actually a layer of tissue or special sheath cells called the Endodermis. So dermis tends to mean skin, endodermis tends to mean within, so endo means within. Epi, by the way, if you see epi, tends to mean upon or on top. So endo is within, and dermis means skin. So it tends to mean the layer of skin around the plant. It's technically kind of like a skin. Underneath that is the endodermis, and this also contains the ability to pass water to the xylem vessels. And just under the endodermis is a layer of meristem cells, just like you found in the stem. And of course, the meristem cells can divide into either xylem or phloem. So if the exam shows you a diagram of either a cross-section of a root or of a stem, you should be comfortable labelling the xylem, the phloem, the cambium in the stem, and also explain the function of the meristem cells, which are to divide into new xylem or phloem cells for vessels. So this diagram shows you the diagram of a leaf, and we can see in the leaf then actually, and I'll zoom in a bit, the phloem and the xylem are here. And as is the repeating pattern you've seen so far, the xylem is inside and it's marked here in blue, and the phloem is on the outside here. It's a kind of lightish colour just after the red there. Now you have to imagine this is actually a kind of what we call a transverse section of a leaf, so this would be sort of a, if you cut the leaf in half, that part there going along the top is actually the surface of the leaf, and here is the bulge underneath the leaf, and it's showing you kind of a, like looking through a tunnel of the midrib, the big thick rib in a dichotomous leaf. And of course you also see these tiny small blue bits there, and that's the vein, the smaller veins that join the midrib somewhere towards where you are standing. So those veins that you see in the plant, those kind of bulges, if I draw a really badly drawn leaf there, those veins there, which kind of spread out like that, the diagram you're seeing, you're actually seeing kind of a, if I cut the leaf in half like so, and turn one half to face me, you're actually seeing a bit of the midrib there, and also parts of the veins. And the veins, of course, are made up of xylem and phloem, and that's why they're veins. They're vessels or vascular bundles, and they carry all the things the plant needs to transport. Now, a dicotyledonous leaf tends to have one central midrib there, or midrib, sorry, and then branches of ribs, or I keep saying ribs, branches of ribs that become smaller and smaller and smaller as they divide. And that's a technical feature of a dicotyledonous leaf, or dicotyledon. And in the veins we tend to say that the xylem is usually on top of the phloem, so there's a phloem underneath, a xylem on top. Or of course you could say it's more central, the xylem. So you should now be quite comfortable using diagrams or drawings or photographs of the stem, the leaf and the roots, and identifying the xylem and the phloem, and of course in some cases the cambium, and understanding the myristoms themselves are found in the cambium, and they can differentiate into xylem and phloem. You should also know that the xylem carries water and dissolved dissolved minerals, and the phloem tends to carry sugars such as sucrose. Now we're going to go into more detail now. Since we've completed objective 1, we'll now go into detail about the structure of actual individual xylem vessels, sieve tube elements, and companion cells. And we'll look at the function of these three things too. So this diagram shows you a zoomed in cross section of a vascular bundle, and the red bits there are xylem vessels. You see one there, one there, one there, and so on. And it's actually made of, first of all, an empty lumen. So this lumen, a bit like a vein or an artery, has nothing inside it, and like the artery carries blood through the lumen, the xylem carries water and dissolved minerals. And around the xylem is actually what we call a thick lignified wall. So basically, it's lignified walls, are walls that are impregnated by a chemical called lignin. And as the xylem grows and it becomes lignified, the lignin creates a waterproofing effect of the walls of the cells. And because this lignin impregnates the walls of the cells, what it means is the cells themselves actually die. And the contents decay, and this leaves behind a dead structure of a xylem vessel. And of course the walls are now waterproofed. The old cells that were once living have died, and their outsides, their walls, have now become waterproofed by the lignin impregnated in them. And of course that decay leaves the walls behind, and the rest of the cell completely breaks down, and that gives you this kind of straw-like empty tube, very strong tube, and this strengthened tube is open at all times, doesn't collapse because it's quite strong from the lignin, and of course it can carry water. And that's better than having a living system, because living systems tend to shrink when there's no water, whereas with xylem, even when there's no water, the vessels will still stay intact, so the plant has a better chance of survival. And of course the waterproofed sides of the Xylem vessel means that the water will not leak out. But actually, in some places in the Xylem vessel, you will find, and of course there's a vascular bundle, so there's more than one, but you will find there are tiny gaps between one vessel and another because lignification hasn't completely been carried out. So you might find a pit there, a pit there, a pit there, and a pit there. And they're called bordered pits. And those bordered pits allow water to flow through from one vessel into another. So that still can happen, even though most of the wall has been lignified. And of course that also allows water to pass from the Xylem vessels into the living parts of the cell, or of the plant, sorry. So the water can actually go through the xylem and then leave the xylem and enter other cells in the plant, which is obviously important for the transport system, a bit like how you want tissue fluid to flow out of the arteries through capillaries. You also want water to flow out of the xylem. So the bordered pits allow that to happen. Now, in the book, it does tell you some reasons why xylem are adapted to carry water, and some good ones are, the first thing is, xylem vessels are very narrow. Okay, they're very narrow, they're like tiny straws, and this means that when water passes through them, the water molecules have a cohesive force, so they tend to join to each other through small intermolecular bonds, and as the water flows through, it pulls water below it up that tube. And we said before, it's a bit like taking a straw, sipping some water, and then suddenly putting your thumb over the straw and taking the straw out. The water will stick in the tube or the straw, even though gravity is trying to pull it downwards, because the water molecules have a cohesive force. And the thinner the tubes, the thinner the xylem, the more strongly this force is felt. Whereas if you widen the xylem, you'll notice that whilst there'll be a small amount of water on the sides, most of the water will actually fall through. And if gravity allows the water to be pulled down to the roots, the leaves and other parts of the plant cannot gain water. So you need thin tubes, to increase that water column moving through the xylem, and therefore this allows capillary action to take place. I'll move this out of the way now, and we'll carry on. So, next we can talk about the fact that the xylem is made of dead cells, and they're dead because when they become lignified, they are starved essentially of water. We shouldn't usually use the word starved, we could say deprived of water. And those dead cells decay to form a hollow inside, or lumen, and these form a continuous column of the vessel. And we talked about the fact that there's some pits or bordered pits between the xylem vessels caused by incomplete lignification, and therefore some water can move through one vessel and then pass to a different vessel, or even pass out of the vessel to parts of the plant. The lignin is deposited in a spiral shape, or sometimes a reticulate shape, and this generally means that the xylem can stretch and flex as the plant grows, and it also allows the plant branch to bend without destroying the delicate xylem. So it's quite flexible due to the way that the lignin is deposited in the walls of the cell. And of course, water is not impeded or stopped for a number of reasons. First of all, there's no end wall, so you'll find that there's a lumen that's continuous from one side to the other. There's not suddenly some sort of wall at the end blocking the water. You will also notice there's no cell contents, because remember, if the cell dies, these contents decay. There's no nucleus or cytoplasm. These are of course cell contents, and therefore this increases the way that the water can pass through those dead cells. And of course, lignin's thickened to prevent the walls from collapsing, so it provides a solid, strong structure of the xylem. Now the phloem actually is a tissue, and it's made of two types of cells. They have sieve tube elements and companion cells. So sieve tubes, first of all, are not quite cells, as they have hardly any cytoplasm and they don't have a nucleus, and they form a tube. And actually, if I draw you a diagram of the phloem, like so, I can draw a wall going down, another wall going down there, and in between parts of the phloem, you see this kind of what we call cross wall, and that's a side view. But if I draw you a top-down view or a bird's eye view, or we could call it a transverse section view, and usually it's got 5 or 6 sides to the phloem, it's not completely circular, you'll see it looks like this. You'll see tiny holes like a sieve. And these are called sieve tubes or even sieve plates. And actually, they differ to the phloem, because, sorry, it's the xylem, because the xylem doesn't have these cross, whoops, that was running, but these cross walls or sieve plates running down them. The xylem has a complete lumen that's completely empty, that doesn't impede water flow. So I can write down, first of all, that these are called sieve tubes. So I'll write down sieve tubes, a bit like a sieve in your house to separate water from pasta. And of course these are called sieve plates. And we also have a secondary cell attached to the sieve tubes, and they look a bit like this. So you've got the cell there, or a companion cell, and if I take the eraser and quickly just draw a gap there and a gap there, this gap is actually called the plasmodesmata, and it links the cytoplasm, the very small cytoplasm the size of the sieve tube, with the cytoplasm of that companion cell. So those gaps are called plasmodesmata, and of course it links the companion cell to the sieve tube by the cytoplasm. I'll draw that in now. That's it, plasmodesmata. You'll also find that the companion cell has a very large nucleus, so I'll draw that in. And we'll draw it in red. So there's a large nucleus, which I won't label, but you should know it's a nucleus. But also, you will find that these companion cells tend to have these things, which I'm sure you can recognise if I really badly try and draw them, from Mr. Wells' lessons, and they are mitochondria. And I think I've drawn them a bit bigger than they should be. But these mitochondria, as I'm sure and hope you know, are the site of cellular respiration. Now, there's actually a very large number of mitochondria, so I'll just keep drawing them in the sieve tubes, which indicates that a lot of energy is required in the sieve tubes in the form of ATP. Sorry, the companion cells in the form of ATP. So I can draw in using a pen. I'll draw a line to those mitochondria and quickly label them for you. So remember, with cells that have lots of mitochondria, they are usually cells that require lots of energy. So neurons tend to have lots of mitochondria to send nerve impulses, and these companion cells have mitochondria because they obviously require energy too, and we'll find out why later. So the companion cell has a thick layer of cytoplasm. The sieve tubes have a very thin layer of cytoplasm. They're connected by the plasmodesmata, which is a gap between the two cells, and of course there are many mitochondria inside those companion cells to aid in cellular respiration. These are called sieve plates, and they are contained in the sieve tube element of the phloem. I should probably mention that those companion cells run all the way down the phloem. Now, we will go into this in more detail in another video, but I should mention that the sieve tube part allows sucrose to dissolve in water to form sap, so the sieve tubes carry sap, and therefore sucrose and sugar, up and down the plant. Now, the companion cells actually carry out the metabolic processes required by the sieve tube elements using ATP, and this includes helping to load the sieve tubes with sucrose at certain points of the plant. So by now we should be able to describe, with the aid of diagrams and photographs, the structure and function of the xylem vessels, the sieve tube elements, and the companion cells.